Sean kindly asked me to talk about phylogenetic diversity. I'm at the Australian Museum, by the way, not the Australian National Museum, the Australian Museum. I do a lot of typically Australian museum research, that is museum-oriented research. That involves very much a focus on the products that museums produce, including phylogenies, and also the importance of collections and so on. My cute little subtitle here is extracted from Sean's email to me about what I should talk about, and it finishes with, "And any other topics, Dan, that you'd like to bring up?" So with those tight constraints, I'm going to spend a short amount of time. I should say I'm not really per se a geneticist. I've been involved with GeoBon and other groups, Diversitas and others, happily collaborating with geneticists and kind of looking at this fusion between kind of the phylogenetic level of thinking, if you like, and the genetic level of thinking. And hopefully I'll be able to highlight some of those linkages in my talk this morning a bit. This is one of the slides I use if I'm trying to introduce to people or review for them what this phylogenetic diversity PD measure is. This comes from some old work of mine. In fact, PD goes back to 1992, so we're kind of all celebrating next year 25 years for this indicator or index. In the upper slide here, this is actually from a real study on cyclamens affected by climate change in Europe. And so as a quick summary, we don't even need a definition of PD. I can simply say that the amount of PD conserved on that tree in the upper part of the slide is shown by the red amounts. And under their study on climate change, it appeared that at the species level there were large impacts on species, but it was kind of a good newspaper because it said, in fact, the total amount of PD that could persist under climate change could be quite large. And indeed, if you see it, there's a lot of red on that slide. There's a lot of PD remaining after the impacts of projected climate change. Of course, the point I make in some of my papers is that you can be unlucky, and we often are unlucky, and if the distribution of those climate change impacts was clumped on the tree, you could have a situation like the bottom there, where in fact the same number of species has been lost, but the PD remaining, as shown by the red on that slide, is quite small. And this is kind of a pointer to a lot of the research I've been doing recently on corals and other key taxonomic groups, where we looked for those clumped impacts, and those black dots indicate potential tipping points. So again, for us in this meeting, perhaps if we apply these sorts of tree models and PD at the genetics level, we may be interested in these sorts of tipping points where for a given loss of populations, there could be a very large or a very small loss of diversity. And I'll return to that issue. So that's a quick review. Again, kind of Sean was interested in successful measures. I suppose we could say after 25 years, PD since 1992 is successful. That orange part of the graph there shows that since 1992, there have been more than 10,000 papers in Web of Science with phylogenetic diversity as a key word. But science is messy, I've learned. And so it's really been kind of a really interesting history of how an index or an indicator that we propose, say back in 1992, can take on a life of its own and have unanticipated applications. A lot of it has related to biodiversity conservation, certainly, and that was my original intention for the measure. I did not anticipate the use of phylogeny in community ecology. And that has been a little bit of a mixed bag, a little bit of a curse in that ecology has so many diversity indices, but not all of those diversity indices are necessarily biodiversity indices, and that's a point I want to return to. So it's been kind of a really messy, interesting literature going down to that community level. For me, the other part of this I did not anticipate was the explosion in the number of studies in microbial ecology that use PD. And that's really because they don't have good species, in one sense, we can say. So they became some of the best workers in the world, some of the best software and so on, some of the best indices. And this is extended in interesting ways, for example, to human health. So if we're thinking about health indicators, it's now the case, I've done a bit of a meta-analysis on this, and the PD of your own gut microbial community, for example, is very important to your own health. And indeed, there are something like 12 or 15 non-communicable sort of lifestyle diseases where lower microbial PD is associated with poor health. So this is a very interesting, unanticipated development. Maybe that relates to resilience in the sense that Michael was talking about that word resilience, so that if you lose PD in your gut microbial community, you're losing resilience and you are more prone to various ailments and so on. I always say you cannot be too careful with your health, so take Dr. Faith to lunch, and that's probably the best thing you can do. Now, there has not been, to get to the point of this story, there has not been that much work really out of these 10,000 papers and so on since 1992 at the level of genetic diversity. So this immediately is a gap. And that's kind of paradoxical, because if we go back to my 1992 paper, which I rarely do, but if I go back to it, the second example after I looked at global bumblebees in that paper was I looked at this crested newt species, or really it's a super species across Europe, and a mitochondrial DNA tree, and pulled out certain principles for the sorts of metrics that we would like to use if we're working on PD. Important take-home message here as I go through this is that, as I emphasized in that original paper, and yet it's sometimes forgotten, if we have something like PD, we do not have a single index. We have a whole calculus, if you like, a whole set of calculations that we can carry out. And I think this will be an important point if we're developing indicators or indices for genetic diversity. My conjecture is that we do not want a single indicator or index. We actually want a calculus for that. So let's return to that point. So what I showed in that original example was this idea of complementarity, all right, these sorts of marginal gains and losses. In this little example across Europe, the green lines on that tree could notionally represent the amount of genetic diversity, if you like, which could be regarded as secure already for whatever reason. And if we're wanting to set priorities on the important populations across Europe for conservation, then I argued in that paper that, in fact, as shown by the red lines, we could make a gain of 7 to 10 PD units, which are basically counts of features, or in this case, genetic variants, if you like, if we were to protect one or more of the Western European populations, which uniquely represented certain genotypes, mitochondrial types. So that's called complementarity. The length of that red line is the gain we can make in genetic diversity if we take action on those populations. Similarly, as shown by the blue lines, we could make a gain of 9 PD units if we protected the populations in Turkey, uniquely representing types 15 through 17. So this was an important principle. Many people over the 25 years have regarded PD as another index, particularly the ecologists who already have 100 indices of diversity. But in fact, PD is something you would want to maximize as a measure of biodiversity. But the measures that you use for decision making are measures like endemism or complementarity and so on. And that's an important principle.
Sorry,
Endemism then would be, in this case, the question is, what would endemism be? In fact, I use endemism in two ways. The conventional meaning of endemism would be that portion of PD which is unique to a place or an area. But we can also talk, in a sense, about endemism phylogenetically, which says that population 17, for example, on that phylogeny has phylogenetic endemism. It uniquely holds certain features that are held by no other population. So endemism does not, for a mathematician, have to be something which is strictly geographic areas. In fact, it is the unique diversity attributable to an object, in this case, a population. I want to highlight this word distinctiveness, which, as expected, has come up in Michael's talk and several others as this kind of tantalizing way to try to get at the importance of certain areas or certain elements of biodiversity. My recommendation is that we try to sidestep it. Distinctiveness actually has been problematical in the broader biodiversity literature. One reason is that it really is not capturing what we capture when we talk about complementarity. And I think Michael or Sean or both of you highlighted the fact, I think Sean did, the fact that complementarity is this context-dependent thing. It changes, and we want that property. Typically, when we try to quantify distinctiveness, it's regarded as a static score. But if we look, for example, on this diagram at, say, population number 10, it might have been thought to be distinctive, but once we've protected the red and the green and the blue, in fact, the marginal gain, the complementarity value we get if we protect 10 is quite small. So distinctiveness might have been a static score, but in fact it would not be indicative of this changing complementarity that we need to use as a decision maker. All right, so that's a quick look at what complementarity is, and I'm highlighting that for discussion as we look at indices and metrics. So current research was one of the other topics Sean asked me to sort of talk about in terms of PD, and we're looking a lot at IUCN Red List and threatened species in the Tree of Life. This is from OneZoom. If you have never looked at OneZoom, go to www.onezoom.org. It's a fantastic kind of fractal mathematics way at looking at like 50,000 species on a single computer screen, because it kind of expands out. What's the coloring on this diagram? The coloring is the IUCN categories. So red, for example, is critically endangered. Blue is extinct already. Green is, in IUCN speak, least concern, in other words, secure species. So a lot of our work now is about how we can set priorities taking both IUCN categories and the Tree of Life into account. There's been a lot of work on that for the past decade, but believe it or not, we still need to resolve the mathematics, basically, of how we do that properly. Let me give you a peek at this, because I think some of this might be relevant to the priority setting we might do if we use a tree model to understand genetic diversity. So we'll look over here. There's that little red species there. I think this is a group of frogs. And if we were to conserve that critically endangered species, why do you think that might be a high priority? Well, the brown color on that branch is also endangered species. So if you could protect one species along that branch, you are conserving the entire length of that branch. All right? So in fact, it might be millions of years of evolutionary history that could be conserved if you target your conservation dollar on that critically endangered species. So working out that mathematics in terms of probabilities, this goes back to some of Weitzman's early work on expected diversity and so on, is sensible. We can also combine that with what we call risk analysis, which says expected diversity looks at kind of average outcomes, but we want to look at worst case outcomes and so on. So we can do that kind of analysis. I won't go into detail on that now, but again, I'll highlight that as a topic that we may want to talk about. That's kind of the scary news. So we use this in a gallery in the Australian Museum, but we're very careful in that gallery not to have the public only hear these kind of frightening stories about branches falling off the Tree of Life and so on, and species going extinct. We highlight good news stories too. And here's an example of a good news story. And this little branch of the Tree of Life for these frogs, there are many critically endangered species, but notice that one species there with the green leaf. There's a secure species, and we're highlighting these as phylogenetic bright spots. These are the good news stories where, as long as that species persists, it is kind of holding up an entire branch of the Tree of Life. So in addition then to highlighting, as a conservation biologist, our priorities for protecting critically endangered species, we want to give more attention to this, like this tinker frog here, where it's a secure species. It might be neglected and unappreciated because it is secure. And you'll see that many times in the IUCN Red List. The species will be called common, least concern, and it won't get any attention. But these species really need to be appreciated as kind of holding up a branch when all the sister taxa around them are going extinct. Again, is that relevant to genetic diversity? Possibly it's a useful way to think about genetic diversity as well. So why do we care about these branches falling off the tree? And indeed, why do we care about genetic diversity? And I think it was useful this morning in Michael's talk that he looked at the CBD goals and so on. There was kind of a reference to the socioeconomically important species. But my argument as a biodiversity science is that PD, which is an example I'm going to show you of a very general biodiversity framework, is all about living variation, and that variation has variety in the sense of what we call option value, which is a term stolen from economics. It basically means values for future generations, often unanticipated values. So in Australia, we found out just 2 weeks ago that the Tassie devil, one of our endangered species, which is absolutely a wonderful creature, has a potential value to humans because its milk may be a good killer of bacteria which are drug resistant. So here's a totally unanticipated benefit from a species. If someone asks you about the value of variation, that's the ultimate value of variation. It is not the currently perceived so-called socioeconomically important species or genetic variants. It is in fact the importance of maintaining variation for future generations because of these unanticipated gains. My conjecture is that this is one way we want to look at the importance of genetic diversity as well. And there's a balancing act between maintaining genetic diversity for this general option value, sometimes it's called insurance value, and Michael, I think this may relate to resilience value as well. And this may depend upon the geographic scale that we're looking at. This is an important kind of valuation of genetic diversity that we may want to balance or synergize or trade off with the current important values of particular genetic components. So it's not that we're denying those known importance now, but we also at the same time want to maintain variation for future generations. So that's my key point. I'll make also a historical point. In some of my talks, I go into great detail about this, but if you pick up the average paper on biodiversity right now, you don't hear this story enough. And part of the problem is that we're dominated by the ecosystem services story, which imagines that in fact biodiversity is all about intrinsic value, which is this kind of unquantifiable, non-anthropocentric value. But in fact, that's not true. That is a false history that you're hearing in the average paper. The roots of biodiversity, and so I think the roots of genetic diversity, are about truly an anthropocentric value, but it's the most grand value because it's this sort of variation for future generations. So there's an interesting correction of history that I think is important here. So the other thing I think that is relevant to talking about genetic diversity is how we think about biodiversity more generally and what sort of general model we're considering. PD is in fact just one part of a general model. Let's see if I've got a pointer here. The way I think of it is that biodiversity is always quantifying variety. So we have a strict definition. Biodiversity has been defined lots of different ways, particularly among the ecologists. But if we stick to something very similar to a CBD definition, it's really about living variation at multiple levels. And the other point to make is that typically then we have some units that we'd like to count up, but we don't, most of those are unknown to science. So even at the species level, most species, many species, even in Australia, are unknown to science. So typically we're using proxies or surrogates. And the model for that is what I'm talking about here in this framework, where PD is an example where we're using a pattern, indeed a pattern process model, phylogeny, to go from objects, which are species, which we've observed, to making an inference at a lower level, where we'd love to count up all the features or characteristics or genes, but we don't observe those directly. And if we don't, then we can use, this is why, in a sense, PD is important, because it stands in as a proxy for what we would like to count up, but we cannot directly count up. And I think that's relevant to the indices we use for genetic variation. So again, not just species, but populations may be objects. The phylogeny may sometimes be a true tree, but could also be a network. And Min's work in systematic biology has extended it to the network context. And so the units, again, that can be more clearly gene types, if you like, genetic variants. But there are many other aspects to biodiversity where, in fact, we might be using areas or places, environmental space, and other measures. The last point I want to make in this very short talk for discussion is that if we're looking at the sorts of models for quantifying genetic variation and we think something like a tree model might be useful, we have to keep in mind that there are other competing models for this. And so as popular as a network or something like that might be for modeling it, something like this environmental sort of space could be a better model if we want to get at adaptive variation rather than all variation. So let me finish up by looking at what I mean by that. Here's a contrast between the two sorts of models we can look at. The PD model on the left shows a phylogenetic tree, and that basic model is a classic ancestry model. Common ancestry or shared ancestry explains shared features. So the shared features are the black dots. And so shared ancestry explains that. And that's why then PD works mathematically in a way. If we can maximize the amount of PD, the amount of branch length that we can serve, we're maximizing the number of different features that we can serve. But on the right is a totally different model that equally might be plausible and useful for genetic variation. And that's a model where environmental dimensions may account for features. So instead of shared ancestry accounting for shared features, shared habitat or shared environment might account for shared gene types. So that model is interesting, and we're exploring that now a little bit in terms of how we look at genetic variation. The other point to make here, before I go on and look at that a little bit in my last slide, is that this framework will be useful for genetic diversity, whether we use PD or ED or other models and so on, because these are effectively, this is a framework for inferring how we measure variety, and it allows an entire calculus. That idea of a calculus that I talked about is relevant here. So if we take those units, many other measures, and Anne will talk, in fact, in her talk about some of these, particularly Hill numbers, I think. Once you have your units and your ability to infer the units based on some models, it's like opening the box and for free getting 100 other calculations you can make. Endemism measures, dissimilarity measures, complementarity, and so on. Hill numbers that Anne will talk about in her talk. And so we can steal the measures that have been developed over the last century at the conventional species level. And there are hundreds of indices where we use species information. If we're using PD and we make an inference about gene types or features, every measure that has ever been invented at the species level transfers across to the other level. And so we're doing this a lot with PD. We have PD dissimilarities where two areas are compared in terms of the amount of their differences in features effectively. And Anne has done this for Hill numbers, and she'll talk a bit about that. Why is this important? It's important in two ways. It gives you a wealth, a whole calculus working at different levels. So in a way, for genetic diversity, we have an entire calculus simply by taking every measure that's conventionally applied at the species level. And the other thing that's useful about this is it says those things in blue do not have to themselves be called diversity measures or biodiversity measures. Those are other calculations. And if you read the literature right now, the ecologists are in an uproar saying we have so many diversity measures, we can't make sense of it, and isn't it all a mess? Well, one of the reasons it's a mess is they're calling all of those calculations diversity measures. And so no wonder there's confusion. So in a way, this can help sort out, particularly for genetic diversity, what we call a biodiversity measure versus another calculation. So that's the other point there. So just to finish up, in my remaining 2 slides, the way we're looking at this sort of environmental space, these non-tree models genetically, is starting from the microbial context where we have these sorts of 16S trees, commonly in microbial ecology, and we have these lineages, but analyses like multidimensional scaling, ordination analyses, reveal environmental dimensions, which are in fact much better explanations of the patterns, as these black dots are different samples of house dust microbe communities. And we find that if we go to different houses, different seasons, and so on, we can better understand the turnover in genetic diversity using this sort of environmental model. And that probably is a lot better than a tree model. So as much as I like the tree models, I think we need to open the door, and this is probably the case also for genetic diversity, to considering these sorts of environmental spaces as alternative models. So I'll come back to that point and just sum up the conjectures, I suppose, that I think are useful points for discussion. PD is popular, but the genetic diversity links have not been explored much for whatever reason, in spite of the fact that really the earliest studies looked at it as potentially useful for genetic diversity. Tipping points, let's go back. Tipping points are looked at phylogenetically at various scales, and that might be a useful possibility for genetic diversity as well. PD, as I said, is about variety, so it is a biodiversity measure, but it's not that useful as a single index. And PD calculus, all the associated calculations that we can make within the PD framework, are much more useful than PD itself. Our current research at the species level might be relevant to genetic diversity in that we're using probabilities of extinction, we're doing risk analysis, looking at worst case losses and so on. We're also looking not just at bad news, but looking at bright spots. And again, these sorts of good news stories might be useful when we're looking at genetic diversity. So I've talked a bit about this shared calculus and insurance values. Health indicators, MCA there stands for multi-criteria analysis. And I think the point in looking at health indicators here using these sorts of biodiversity measures, it may be the case that we don't want to, as the Ocean Health Index has, simply add up a lot of individual variables and so on, and that's a popular thing to do. But many of these are in competition, so we want to actually look at trade-offs among them and so on, and adding up misses that information. And finally, the last thing I talked about in this brief overview is alternatives to the tree calculus, where we look at environmental space, and we're starting to look at the applicability of that for genetic diversity as well.